Hello students, welcome to our live APS classes, and as all we know that this is our foundation course especially for the students from 9 to 12, and in our today's live lecture we will continue with our chapter number 9, that is biomolecules, and this chapter is for class 11th. And regarding this chapter, today we will discuss important 20 MCQ questions or objective questions from this chapter, and this is the syllabus for the term 1 examination that is to be held in November 2021. Now let us start with our chapter, that is biomolecules. And as all we know that biomolecules are the most important organic molecules. These are the most important organic molecules. So first of all, good evening to all the students who joined with us our live APS classes, and the brief introduction of the chapter, that is the biomolecules. Biomolecules are the most essential organic molecules which are involved in the maintenance and metabolic processes of the living organisms. That means biomolecules, these are most essential organic molecules, and these biomolecules are involved in the maintenance and the metabolic processes that are involved in all the living organisms. So in short, we can say that these are the metabolic processes that are involved in all the living organisms. The non-living molecules are the non-living molecules are the actual foot soldiers of the battle of the substance of life. That means the non-living molecules are the actual foot soldiers of the battle of the substance of life. They range from the small molecules such as primary and secondary metabolites and hormones to large macromolecules like proteins, nucleic acids, carbohydrates, and lipids, etc. That means the non-living molecules are the actual foot soldiers of the battle of the substance of life, and they range from small molecules such as primary and secondary metabolites and hormones to large macromolecules such as proteins, nucleic acids, carbohydrates, and lipids, etc. So this is the brief introduction of the chapter, that is the biomolecules. Now let us start with our first question from the chapter, that is biomolecules, and our first question is: feedback inhibition of enzyme is affected by which of the following? Options are enzymes, substrate, C, end products, and the last one is intermediate end products. That means feedback inhibition of enzyme is affected by which of the following? There are four options. So which option is the right option for the question number 1? That is the feedback inhibition of enzyme is affected by. So the feedback inhibition of enzymes that is affected by the end products. That means the feedback inhibition of enzymes is only affected by the end products, and it has no effect on the enzyme, substrate, as well as intermediate end products. But only the end products are involved in the feedback inhibition of the enzymes, and these end products, they totally affected the feedback inhibition of the enzymes. So here this is the right answer for the question number 1, that is the feedback inhibition of enzyme is affected by the end products. Now shift to the question number 2. An example of competitive inhibition of an enzyme is the inhibition of, options are succinic dehydrogenase by malonic acids, B, cytochrome oxidase by cyanide, C, hexokinase by glucose 6 phosphate, and D is carbonic anhydrase by carbon dioxide. That means an example of the competitive inhibition. Competitive inhibition of an enzyme is the inhibition of either. There are four options. So which is the correct option for the question number 2? An example of competitive inhibition of an enzyme is the inhibition of succinic dehydrogenase by malonic acid. That means an example of competitive inhibition of an enzyme is the inhibition of succinic dehydrogenase by malonic acid, not by cytochrome oxidase by cyanide, not by hexokinase by glucose 6 phosphate, and even not carbonic anhydrase by carbon dioxide. But the example of the competitive inhibition of an enzyme is the inhibition of succinic dehydrogenase by malonic acid. So this is the right answer. Now shift to the question number 3, and your question number 3 is insulin is made up of dash and dash. Options are glucose and fructose. Insulin is the hormone that maintains the sugar level in the body. So insulin is made up of either glucose and fructose, B glucose and fructose, C fructose and mannose, and D is mannose and glucose. So out of these, insulin is made up of dash and dash, and it is your question number 3. So insulin is made up of glucose and fructose. So insulin is made up of glucose and fructose. Glucose and fructose. So these are two by which insulin is made up of, and that are glucose and fructose. Not fructose and mannose, and not mannose and glucose, but only by the glucose and fructose insulin is made up of. So here this is the right answer. Glucose and fructose. Now shift to the question number 4. Enzyme increases the rate of reaction by. That means which enzymes increases the rate of the reaction? Either lowering activation energy, B increasing activation energy, C increasing temperature and pH, and D is decreasing temperature and pH. That means enzymes increases the rate of reaction either by lowering the activation energy, by increasing the activation energy, by increasing temperature and pH, and the last one is decreasing temperature and pH. So how the enzyme increases the rate of reaction, and it is your question number 4. अब इसमें आपको मैंने बताया था कि insulin is made up of glucose and fructose. So glucose and fructose are those by which insulin is made of, because insulin is polysaccharide. Insulin is polysaccharide. Insulin is polysaccharide, and by this it is made up of glucose and fructose. So glucose and fructose. So insulin is a polysaccharide and it is made up of glucose and fructose. So by this it is made up of glucose and fructose because insulin is polysaccharide. Enzyme increases the rate of reaction by. So enzyme increases the rate of reaction by lowering the activation energy. By lowering the activation energy. So this is the only factor by which the enzyme increases the rate of reaction, and that factor is lowering the activation energy. Lowering the activation energy. So हरी बेटे जल्दी से आप इसका आंसर बताएं Key enzymes that increases the rate of reaction either by lowering the activation energy, because enzymes increases the rate of reaction by decreasing the activation energy, because enzymes increases the rate of reaction, enzymes increases the rate of reaction by decreasing the activation energy. Enzymes increases the rate of reaction by decreasing the activation energy. This is the explanation. Decreasing the activation energy. So this is the explanation by which we can say that enzymes increases the rate of reaction by lowering the activation energy, because enzymes increases the rate of reaction by decreasing the activation energy. So this is the explanation for the question number 4, that is enzymes increases the rate of reaction by lowering the activation energy. So this is the right answer. Now shift to the question number 5. Which of the following statements regarding enzyme inhibition is correct? Options are: competitive inhibition is seen when a substrate competes with an enzyme for binding to an inhibitor protein. Second, non-competitive inhibitors often binds to the enzymes irreversibly. C. Competitive inhibition is seen when the substrate and the inhibitor compete for the active site on the enzyme. And D is non-competitive inhibition of an enzyme can be overcome by adding large amount of substrate. So there are 4 options for the question number 5, and your question is which of the following statements regarding enzymes inhibition is correct? तो इनमें से कौन से statements हैं that are correct regarding the enzyme inhibition? So बेटे जल्दी से आप इसका आंसर बताएं क्वेश्चन नंबर 5 का. हरिए बेटे. So बेटे, out of these following statements regarding the enzyme inhibition, and the statement that is correct is competitive inhibition is seen when the substrate and the inhibitor compare for the active sites on the enzymes. So this is the correct statement for the enzyme inhibition, and the correct statement is competitive inhibition is seen when the substrate and the inhibitor, both are compete for the active site of an enzyme. That means when both the substrate and the inhibitor compete for the active site of an enzyme. So this is the correct statement for the enzyme inhibition. So this is the right answer. That is, competitive inhibition is seen when both the substrate and the inhibitor compete for the active site of an enzyme. Next is the question number 6. Which of the following is not pyrimidine? Which of the following is not pyrimidine? Options are uracil, cytosine, guanine, and thymine. So you have to choose out of these which is not a pyrimidine. Options are uracil, cytosine, guanine, and thymine. So guanine is not a pyrimidine. So this is the right answer. That is, guanine is not a pyrimidine. Next is your question number 7. Hydrolysis of starch occurs with the help of. Options are peptidase, B amylase, C sucrose, and D is lipase. So hydrolysis of starch occurs with the help of. Options are peptidase, amylase, sucrose, and lipase. So it is the question number 7. hydrolysis of starch occurs with the help of. That means hydrolysis of starch occurs with the help of बच्चे। अभी इससे पहले हमने बताया कि इनमें से कौन है जो पिरामिडिन नहीं है, तो हमने कहा कि ग्वाइनिन इज नॉट अ पिरामिडिन बिकॉज ग्वाइनिन इज ए प्यूरिन. So here guanine is not a pyrimidine. Hydrolysis of starch occurs with the help of. Options are peptidase, amylase, sucrose, lipase. So here hydrolysis of starch occurs with the help of amylase. Hydrolysis of starch occurs with the help of amylase. With the help of amylase. So this is the right answer. So hydrolysis of starch occurs with the help of amylase, not peptidase, not sucrose, and not lipase, but only with the help of amylase hydrolysis of starch occurs. Next is your question number 8. Assertion archidonic acid is an unsaturated fatty acid. The reason there are one or more double bonds between the carbon atoms in unsaturated fatty acids. Options are both assertion and reason are true, and reason is the correct explanation of the assertion. Second, both assertion and reason are true, but the reason is not correct explanation of assertion. C, assertion is true, but reason is false. D, both assertion and reason are false. So here assertion, archidonic acid is an unsaturated fatty acid. Reason, there are one or more double bonds between the carbon atoms in unsaturated fatty acids. Options are both assertion and reason are true, and reason is the correct explanation of the assertion. Second, both assertion and reason are true, but the reason is not the correct explanation of assertion. Assertion is true, but reason is false. Both assertion and reason are false. So it is your question number 8. Assertion, archidonic acid is an unsaturated fatty acid. Reason, there are one or more double bonds between the carbon atoms in the unsaturated fatty acids. So here both assertion and reason, both assertion and reason are false. This is the right answer. Both assertion and reason are false. So this is the right answer for the question number 8. Next is the question number 9. Which of the following influence feedback inhibition of enzyme? Options are end product, external factors, enzyme, substrate. So which of the following influence feedback inhibition of enzyme? Options are end product, external factors, enzymes, and substrates. And it is the question number 9. Which of the following influence feedback inhibition of enzyme? So the feedback inhibition of enzyme, that is the end product, not the external factors, not the enzyme, not the substrate, but end product is the following influence feedback inhibition of an enzyme. So this is the right answer. Next is the question number 10. Which of the following is not a polysaccharide? Which of the following is not a polysaccharide? Options are lactose, B, starch, C, glycogen, and D is dextrin. So which of the following is not a polysaccharide? It is your question number 10. Options are lactose, starch, glycogen, dextrin. So out of these, starch, glycogen and dextrin, they are not, so they are related with the polysaccharides, but out of these that is not related with the polysaccharide is lactose. Lactose is not a polysaccharide. Polysaccharide. Lactose is not a polysaccharide. This is the right answer for the question number 10. How can you say that lactose is not a polysaccharide? Because lactose is disaccharide. Lactose is disaccharide. Lactose is disaccharide. And this disaccharide, we can say that out of these, lactose is not a polysaccharide because lactose is disaccharide and it is made up of glucose and fructose. Made up of glucose and fructose. So on the behalf of that reason, we can say that lactose is not a polysaccharide, but lactose is disaccharide and it is made up of glucose and fructose. Next is your question number 11, and your question number 11 is insulin is made up of dash and dash. Options are glucose and fructose, fructose and mannose, and D is mannose and glucose. So insulin is made up of glucose and fructose. Insulin is made up of glucose and fructose. So here option, this is the right answer. That is, insulin is made up of glucose and fructose. And it is your question number 11. Glucose and fructose. Because insulin is polysaccharide. Insulin is polysaccharide and it is made up of glucose and fructose. And it is made up of glucose and fructose. So here insulin is made up of glucose and fructose because insulin is polysaccharide and it is made up of glucose and fructose. So here this is the right answer. Now shift to the question number 12. The structure shown above is, options are mannose, B fructose, C glucose, and D is galactose. So here this is the structure of either mannose, fructose, glucose and galactose. And it is your question number 12. One, two, three, 4, 5, 6. That is 3, 6. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. H12. 1, 2, 3, 4, 5, 6. This is H12O6. So here this is the structure of glucose. Here this is the structure of glucose and it is your question number 12. So this is the structure of glucose and the formula of glucose is C6H12O6. So here the structure shown above is glucose. So here this is the right answer. Now shift to the question number 13. A protein having both structural and enzymatic traits is, options are collagen, B. Trypsin, C. Myosin, and D is actin. A protein having both structural and enzymatic traits is either collagen, trypsin, myosin, and actin. So which is the right answer for the question number 13? हरिए बेटे जल्दी से आप इसका आंसर दें. A protein having both structural and enzymatic traits is not collagen, not trypsin, not actin, but a protein having both structural and enzymatic traits is myosin. This is the right answer. That is, a protein having both structural and enzymatic traits is myosin. Now shift to the question number 14. NADP contain vitamin B1, B2, B3 and B12. NADP contain vitamin. Options are B1, B2, B3 and B12, and it is the question number 14. So NADP contains the vitamin that is not B1, not B2, not B12, but NADP contains vitamin that is B3. So NADP contains only vitamin B3, not vitamin B1, not vitamin B2, and even not vitamin B12, but only NADP contains the vitamin B3. So here this is the right answer. Because B3 is also named as niacin. Because B3 is also named as niacin. Because B3 also named as niacin. So NADP contains the vitamin that is B3 because B3 is also named as niacin. So this is the right answer. Next is the question number 15. With reference to enzymes, which one of the following statements is true? Options are apoenzyme is equal to holoenzyme plus coenzyme. Holoenzyme is equal to apoenzyme plus coenzyme. C. Coenzyme is equal to apoenzyme plus holoenzyme. C. Holoenzyme is equal to coenzyme minus apoenzyme. So with reference to the only enzymes, which one of the following statements is true? And it is the question number 15. Yes बेटे जल्दी से आप इसका आंसर दें. With reference to the enzymes and with the reference of the enzymes, the statement that is the true statement is holoenzyme is equal to apoenzyme plus coenzyme. So with reference to the enzymes, the statement that is the true statement is holoenzyme is equal to apoenzyme plus coenzyme. So this is the true statement with reference to the enzymes. Next is the question number 16. Inhibitor binds to the active sites of the enzymes, hence blocking the reaction. This is an example of option is allosteric inhibition, B. feedback inhibition, C. uncompetitive inhibition, and D one is competitive inhibition. So inhibitor binds to the active site of an enzyme, hence blocking the reaction. This is an example of either allosteric inhibition, feedback inhibition, uncompetitive inhibition, and the last one is competitive inhibition. And it is the question number 16. जल्दी से बेटे आप इसका आंसर दें. चलो मैं देख रही हूँ अगर कोई बच्चा इसका आंसर देना चाहता है जो भी हमारे साथ एपीएस लाइव क्लासेस के साथ जुड़े हुए हैं. So beta, the inhibitor binds to the active site of the enzymes, hence blocking the reaction. So this is an example of the competitive inhibition. Competitive inhibition. Competitive inhibition. This is an example of the competitive inhibition. Because when substrate binds to the active site, when substrate binds to the active site of the enzyme, when substrate binds to the active site of an enzyme, then it completes the reaction. It completes the reaction. When inhibitor binds to the active site of an enzyme, it blocks the reaction. So here beta, inhibitor binds to the active site of an enzyme, hence blocking the reaction. So this is an example of competitive inhibition. Because when a substrate binds to the active site of an enzyme, it completes the reaction. But when inhibitor binds to the active site of an enzyme, it blocks the reaction. So here, this is an example of the competitive inhibition because inhibitor binds to the active site of an enzyme, hence blocking the reaction. So this is an example of competitive inhibition because when substrate binds to the active site of an enzyme, it completes the reaction. But when inhibitor binds to the active site of an enzyme, it blocks the reaction. Next is the question number 17. The fastest enzyme known as, option A is DNA polymerase, B carbonic anhydrase, C carbonic dehydrogenase, and D is DNA ligase. So the fastest enzyme known as DNA polymerase, carbonic anhydrase, carbonic dehydrogenase, and DNA ligase. So beta, it is your question number 17. जल्दी से आप इसका आंसर दें. So the fastest enzyme known as carbonic anhydrase. So this is the fastest enzyme that is known as carbonic anhydrase. So how can you say that the fastest enzyme known as carbonic एनहाइड्रेज? तो बेटे आप इसके थोड़ा सा आप इसका आंसर समझ लें कि क्यों फास्टेस्ट एंजाइम जो है उसको हम कार्बोनिक एनहाइड्रेज बोलते हैं. बिकॉज द फास्टेस्ट एंजाइम known as carbonic anhydrase, it converts 106 molecules of carbon dioxide molecules per second. So the fastest enzyme known as carbonic anhydrase, because though the fastest enzyme that is known as carbonic anhydrase, because it converts 106 molecules of carbon dioxide molecules per second. So that's why the fastest enzyme is known as carbonic anhydrase, because it converts 106 molecules of carbon dioxide per second. So this is the right answer, that is the carbonic anhydrase. Next is the question number 18. Lecithin is a, option is polysaccharide, B protein, C nucleic acid, and D is lipid. So lecithin is a polysaccharide, protein, nucleic acid, and lipids. So beta, it is your question number 18. Yes बेटे, जल्दी से आप इसका आंसर दें क्वेश्चन नंबर 18 का. Lecithin is not a polysaccharide, not protein, not nucleic acid, but lecithin is a lipid, because lecithin is phospholipid. This is the explanation that lecithin is a lipid. Lecithin is a lipid, but it is phospholipid. This is the right answer. Next is your question number 19, and your question number 19 is the minimum amount of energy required to initiate a chemical reaction is called. Options are enzymatic energy, B activation energy, C substrate energy, and D is initiation energy. So the minimum amount of energy required to initiate a chemical reaction, that is called as either enzymatic energy, activation energy, substrate energy, or initiation energy, and it is the question number 19. So the minimum amount of energy required to initiate a chemical reaction is known as activation energy. Activation energy. So activation energy is that energy in which the minimum amount of energy required to initiate a chemical reaction, that is known as activation energy. So the minimum amount of energy required to initiate a chemical reaction is called as activation energy, because in activation energy, the minimum amount of energy required to initiate a chemical reaction. So here this is the right answer, that is the activation energy. And activation energy is that energy that is the minimum amount of energy required to initiate a chemical reaction. Initiate a chemical reaction is known as activation energy. Now it is our last question for our today's live video lecture, that is question number 20. And your question number 20 is enzymes, vitamins, and hormones are common in. Option is enhancing oxidative metabolism, B being synthesized in the body of organisms, C being proteinaceous, and D is regulating metabolism. So enzymes, vitamins, and hormones, they are common in enhancing the oxidative metabolism, being synthesized in the body of the organisms, being proteinaceous, and regulating metabolism. And it is the question number 20, and it is our last question for our today's live video lecture. So हरिए बेटे जल्दी से आप इसका आंसर दें क्वेश्चन नंबर 20 का. So enzymes, vitamins, and hormones are both common in, that is the enzymes, vitamins, and hormones, they are common in regulating metabolism. That means enzymes, vitamins, and hormones all are involved in regulating metabolism, but all are not involved in enhancing oxidative metabolism, not being synthesized in the body of the organisms, and they are not common in being proteinaceous, but all of them are common in regulating the metabolism, or the common which are involved in regulating the metabolism are enzymes, vitamins, and hormones. All are involved in regulating metabolism. So this is the right answer for the question number 20. So here beta we complete important 20 MCQ questions or objective questions from the chapter number 9, that is biomolecules, and this is our chapter number 9 for class 11. So this is all about for our today's live video lecture. So all the students who joined with us our live APS classes, so good evening to all.